So, hello everybody, and I would like to first of all say welcome to Jamal. I'm very thrilled to present Jamal and his company Elicera. They got a very interesting pipeline within cancer, and of course, I would like to say welcome to the audience as well, and hope that you find the presentation very interesting. And please ask questions in the chat. First of all, Jamal will present the coming stock listing. So I hand over the word to Jamal. Welcome, Jamal.
Thank you so much, and thank you for this opportunity. So my name is Jamal Mosle, and I'm the CEO of Elicera Therapeutics. First, very short about the company. It's a cell and gene therapy company focusing on immuno-oncology. And that means that we develop drug candidates that uses the patient's own immune system to fight cancer. And we do this based on CAR T cells and oncolytic viruses, which are two up-and-coming hot new areas, you can say, within this field. The company is based on many years of research performed by Professor Magnus Sand and his group at Uppsala University here in Sweden. And they've been very successful throughout the years in securing research grants and also a large crowdfunding campaign that financed our ongoing clinical phase 1/2 study that I will mention more about shortly. So this has enabled us to advance our projects quite far in academia. So we haven't had to turn to investors until first time last year, when we funded the company with 30 million Swedish kronor in an oversubscribed share issue. And we are currently have an ongoing IPO process. The subscription period actually ends tomorrow. You can subscribe from Sweden and from Finland. So if you're interested in the share outside of Sweden and Finland, then of course it's possible to buy the share as usual once trading starts on June 11th. We have 4 drug candidates under development, 2 CAR T cells and 2 oncolytic viruses. We also have a fully developed technology platform, which we call iTank, which can be used to optimize CAR T cells, but which we also use to optimize next generation oncolytic virus in a project we call ELC201. I will talk more about the business strategy at the end of this presentation, but I can just mention here that our ambition isn't to take a product all the way to the market on our own, but rather to minimize risk and add value to the projects by reaching proof of concept in clinical studies before finding a partner that can take over late-stage development and market the candidates. We see a broad potential in 2 of the products that we're developing, both CAR T cells and oncolytic viruses, and we have 2 more niched projects also. I will describe them shortly. The iTank platform, there's actually a possibility here to out-license this technology platform on its own as a standalone technology to any company basically that develops CAR T cells. So we can use this to gene modify CAR T cells to give them an ability to also trigger an immune response by CD8 positive killer T cells. So this means that both the CAR T cells and the oncolytic viruses, they elicit double mode of action. So you get an attack by the CAR T cell and the oncolytic virus, but we also trigger the immune system by CD8 positive T cells. So you have multiple mode of actions in one therapy. We have relevant experience within both board and management. So Professor Magnus Sand, which is the co-founder and chief scientific officer of Elicera, he was actually part of the very first clinical trial with a CAR T cell in the whole of Europe. This, of course, means that it minimizes the risk that we won't be able to perform the clinical studies that we've planned in the most optimal way. Myself, I have over 13 years of experience from running biotech companies. Among others, I was acting co-founder and CEO of another immuno-oncology company in Sweden called Immunicum. They develop slightly different types of products, but it's still within the same field of immuno-oncology. And we also have a board with broad and complementary competencies. And our chairman of the board, Agnete Edberg, was actually my chairman of the board of Immunicum for many years. So we have an ongoing collaboration, and we know each other since many years back. So there's a good collaboration and foundation with her. But more about the board later.
Good.
So short about the company history, you see Magnus and Dee, the co-founders of the company, to the picture here on the far left. They developed an oncolytic virus between 2007 and 2011 successfully preclinically. They basically took the project all the way so it was ready to enter patient studies, but they lacked the funding necessary to start a clinical trial. So they had to pause the development of this project. Around this time, as a coincidence, Magnus was contacted by a British freelance journalist called Alexander Masters. He had a friend that had gotten neuroendocrine tumors, and he didn't have any treatment alternatives. So the journalist took to internet and searched for interesting treatments under development and clinical trials in this specific indication. And Magnus, since he had developed this oncolytic virus in neuroendocrine tumors and had reported interesting preclinical data, the journalist then contacted Magnus and asked him if there wasn't a clinical trial that he could participate in, his friend. And they started a discussion around the problems that is often relevant, at least in Sweden, where you have a lot of interesting innovations in academia, but that don't always reach the patient since there's a lack of funding to start clinical trials. So he ended up writing an article that got spread in Financial Times and The Guardian and The Telegraph and other news channels. And this ultimately led to a large crowdfunding campaign where thousands of people throughout the world contributed with different sums of money to help this project reach clinical trials. And a large donation was given by Vince Hamilton. He was a businessman that had himself gotten neuroendocrine tumors. Unfortunately, he passed away before he could have an opportunity to participate in a clinical trial, but his family set up a foundation called the Victory Net Foundation that finances research and development in this specific indication, and that also then financed the initiation and the completion then of the phase 1/2 study that is currently ongoing. So Magnus and Dee, they formed Virex, which Elicera was named before in 2014. And they did it not to run it as a company, but basically to hold the assets, the patent applications and the approved patents. And I met Magnus and Dee when I was CEO of Immunicum in 2014. And Immunicum then bought a patent from Magnus and Dee around an adenovirus vector that Immunicum wanted to use for one of their products, but that Magnus and Dee used for oncolytic treatment of neuroendocrine tumors. And since they had secured this funding for the clinical trial, they of course wanted to complete the development of that specific project in that indication. So Immunicum gave Magnus and Dee an exclusive global license to use that technology for neuroendocrine tumors and oncolytic treatment. The study was initiated in 2016, and Magnus and Dee also then applied for a patent for the ITANK platform, which I will mention shortly, which can be used to optimize CAR T cells. Throughout 2018 and 19, they received several large research grants. And I left Immunicum around the turn of 2016-17. And we've kept in touch throughout the years, me, Magnus, and Dee. And I contacted them and we discussed a potential collaboration. We concluded that they had a lot of interesting projects under development, but that they could also, around that time in 16-17, they could continue to be developed in academia for a couple of years since they had research grants and the development could be conducted at Uppsala University. So we picked up discussions again in 2019 and 20, and then we launched Elicera. So we changed the name of Virex to Elicera. We recruited a competent board. We applied for a patent also for ELC401, which is our CAR T cell in solid tumors. Which brings me to the product pipeline. So you see that we have 4 products under development. ELC100 and ELC201 are our oncolytic viruses under development. And the most advanced is ELC100 in neuroendocrine tumors, halfway into a phase 1/2 study in neuroendocrine tumors. ELC201 is what we call next generation oncolytic virus. And the reason we call it that is because we have genetically modified this virus so that we can elicit a combination of 3 mode of actions in one drug candidate. It's also applicable to most tumors, while ELC-100 is restricted to neuroendocrine tumors. ELC-301 and ELC-401 are our CAR T cells under development. 301 is in liquid tumors, and 401 is not in all solid tumors, but in a range of different solid tumors. So we are planning to initiate the clinical trial starting second half of next year in B-cell lymphoma with 301. And then 401, our primary indication is glioblastoma multiforme, which is an aggressive type of brain tumor. So the target that we're going for, we know is overexpressed on brain tumor, but we also think that it's overexpressed on a number of other solid tumors, like melanoma, ovarian cancer, pancreas cancer, and colon cancer. And then, of course, as I mentioned before, the ITANC platform technology that is fully developed and that we think we can use not only to optimize our own products, but also the CAR T cells of other companies. So this should be a platform that's relevant and interesting for any developer of CAR T cells throughout the world. So more about this shortly. So what is an oncolytic virus? It's a virus that you have genetically modified so that it selectively infiltrates and replicates only in cancer cells, but not in healthy cells. So it leaves the healthy cells alone. And when it replicates in a cancer cell, it does so in such a fashion that the cancer cell ultimately bursts, and you get a tumor cell death and an oncolysis. And this is a process that in itself is immune activating. So this will trigger the activation of the patient's own T cells, which will, in parallel to the virus continuing to attack cancer cells, the T cells will also attack cancer cells. You get a double attack on cancer, you can say. And as I mentioned before, the difference between ELC-100 and 201 is ELC-100 is restricted to neuroendocrine tumors and gives rise to these 2 mode of actions that I describe here in this figure, while 201 is applicable to most cancer indications. And we have genetically modified it also with the ITANC platform and another gene modification to elicit 3 mode of actions in one product. You see a picture here of the first patient that we treated with ELC-100 in 2016, and also a picture of Vince Hamilton on the picture below there. We named this product after Vince, since he donated a large sum and made it possible for us to reach patients. So ELC-100 we also call AdVince. It's a clinical study that is done in 2 steps. So the step 1 is a so-called dose escalation study, where the ambition is to determine the safety of this treatment and also find maximum tolerable dose to be treated in an additional 12 patients. So far, we've treated 7 patients, and we do this study together with Uppsala University, who is the sponsor, and they've also already reported promising efficacy data from one of the patients, which is very encouraging. CAR T cells. So what is a CAR T cell? It's a new type of therapy, you can say, that was named Advance of the Year in 2018 by ASCO, which is one of the largest cancer organizations in the world. And they named CAR T cell therapy Advance of the Year since it turned out that this type of therapy has the potential to completely eradicate all tumors in most patients with different types of blood cancers that were previously very difficult to treat and that had undergone standard treatment and had no other treatment alternatives. So they could be more or less cured with CAR T cell therapy. And what a CAR T cell therapy is basically you take out blood from the patient, you filter out the T cells, and then you genetically modify the T cells so that they express a chimeric antigen receptor, the CAR. And this molecule then recognizes a certain target on the tumor cell. And then you expand these CAR T cells in vitro and you re-inject them into the patient. And then the idea is that these CAR T cells will find all the tumor cells that has these targets and then bind into them and start attacking them. And as I mentioned, this works very well in different types of liquid tumors, like B-cell lymphoma and leukemia. But so far, there has been a lot of challenges in getting the same efficacy in solid tumors. And the main reasons for this is that a solid tumor has a very hostile immunosuppressive tumor microenvironment. So the tumor has different ways of hiding from the immune system and also suppressing the immune system. So when the CAR T cell is about to enter a solid tumor and attack it, the environment in a solid tumor actually inhibits the effect of the CAR T cell. Another big issue is that you have very good targets in blood cancer, mainly CD19 and also CD20, which is overexpressed on different types of liquid tumors, but not so much on healthy cells. But you don't have the same good relevant targets in solid tumors. So there's an antigen heterogeneity that you have to take into account when targeting solid tumors. So basically, you need CAR T cells that are able to survive in the hostile tumor microenvironment, and they can also trigger an attack on CAR antigen negative tumor cells. And this is precisely what we believe that we can do with our CAR T cells under development, but that we believe we can help other companies to do with our ITANC platform. So this platform can be used to optimize other companies' CAR T cells under development and make them able to meet these challenges, number 1 and 2 here. So when I started at Elicera, I wanted to, it was of course important for myself to put Elicera and our CAR T cells in perspective and place it on the map of all other CAR T cells that are under development, because we're not alone in developing CAR T cells. There are roughly over 100 companies throughout the world that develop CAR T cells. So where do we fit in the landscape of different CAR T cells under development? And first thing you can mention is that CAR T cells have been developed in different generations. So first generation CAR T cells, they had an insufficient proliferation, they had a brief T cell survival, and they were insufficient immune activation. So due to this reason, they optimized them in different generations, for example, by adding co-stimulatory domains in second generation, another one in third generation. And then you have fourth generation CAR T cells that differ in an important way from the others. And a fourth generation CAR T cell is gene modified so that it contains a cytokine transgene. And this transgene then codes for usually one cytokine. And a cytokine, you can say, is like an immune stimulating factor. So you use this cytokine to trigger a parallel immune response by CD8 positive T cells, usually. And basically, we do the same thing, but instead of coding for a cytokine, our CAR T cells, through the ITANC platform, codes for, it stands for NAP here, neutrophil activating protein, from Helicobacter pylori. So when this is activated, it releases, it starts a process that releases multiple chemokines and cytokines, not just one, but over 20 of them, which we believe together give a more optimal and more robust immune activation against cancer. So this is the way we differ from not only first, second, and third, but also other fourth generation CAR T cells under development. We can also mention that most of the CAR T cells under development today are still second generation, as much as 70%, and only 16% are fourth generation. And of these, most of them are actually developed in academia. So we at Elicera, we've only identified one other company developing fourth generation CAR T cells. And it's a Japanese company called Noile Immune Biotech that has developed a similar technology platform that they call Prime T. And it's interesting because if you look at their news flow the last couple of years, you see that they've been able to secure several licensing deals and collaboration agreements with big pharma companies and larger biotechs that develop CAR T cells. And the rationale is, of course, as I said before, that if you have this type of platform technology, then it should be interesting for other CAR T cell developers, since it can be used to strengthen and optimize the effect of their products. So it's also what we are after at Elicera. So I won't go into the specifics of the mode of action of the ITANC platform, but just to highlight an important point here so that it's not missed. Usually a CAR T cell, it's used alone, on its own. You have the CAR T cell that is directed toward one specific target on the tumor cell. Sometimes it's two. In blood cancer, it's usually CD19, sometimes CD20. In solid tumors, it's other targets. But the point is that the anti-cancer effect you get from the CAR T cell attacking the tumor cell through this one target. Now, the patients sometimes also relapse, even if they respond very, very well to CAR T cell treatment in liquid tumors. These patients, even if you can remove all of the tumor cells, many of them still relapse, and when the tumor comes back, it usually lacks this target. So this means that you can't continue to treat them with CAR T cells because the target that the CAR T cell is going after isn't there anymore. And the ITANC platform, what it does is that we trigger the patient's own CD8 positive killer T cells against the whole set of different relevant tumor antigens on the tumor cell. That's why we have different colors here on, as you see, we have green, white, and red to exemplify that we don't just target the same antigen as the CAR T cell. So you get a broader attack on cancer cell, you get a better capability to withstand the immunosuppressive microenvironment in the tumor, since this whole process makes the tumor very immunogenic. So you change this hostile microenvironment and you make it actually pro-inflammatory and something that helps the T cell to function, the CAR T cell. And you get an immune response that remains even if the tumor loses the target antigen of the CAR T cell. Now, of course, we have quite extensive preclinical data proving this in different ways. And I can't show you everything since we have submitted it for publication. But I want to mention one of the data points that we have here. So we've conducted mice studies where we injected mice with CD19 negative tumor cells and CD19 positive tumor cells, so 50-50. And then we treated them with conventional CD19 targeting CAR T cells, and then CD19 CAR T cells that have been optimized with the ITANC platform. You see it here in red. And as I mentioned before, if you treat a tumor that lacks the CD19 target with a CD19 CAR T cell, it shouldn't have any effect. And despite this, we see a tumor reduction and a prolongation of the survival of these mice when they are treated with our CAR T cells compared with conventional CAR T cells. What we also did was that we extracted T cells from these mice, and then we checked the reactivity of the T cells against CD19 negative tumor cells. And the only T cells that were able to target CD19 negative tumor cells were the ones that came from the mice that were treated with our CAR T cells. So this indicates that the ITANC platform is able to generate this second mode of action through the patient's own killer T cells. As I mentioned before, roughly a little bit over 100 companies developing CAR T cells throughout the world. It's a rough 50-50 split between China and US. You have a little bit more companies in China developing CAR T cells. It's actually an area that is very much almost exploding there, you can say. Not so many companies in Europe so far, only 14 companies, and only one in Sweden, being Elicera. Many of them are still in quite early development. So we have 5 approved CAR T cells today, but all of them are approved in different types of liquid tumors. None have so far been approved in solid tumors due to the challenges that I mentioned before. In China, as much as 85% of CAR T cells under development are targeting CD19. And looking at the whole picture, 50% of CAR T cells under development target the same target, CD19. So this is to keep in mind when we continue discussing Elicera and our projects under development. I write here that we have a large interest in CAR T cells, but still a relatively young field. And what I mean with this is that it's only 5% of CAR T cells under development that are in phase 2 or later. So 95%, which is, of course, almost all of them are in earlier than phase 2 development. So we feel that Elicera has a good chance to really make a difference in the field of CAR T cells, not only with our drug candidates, ELC301 and 401, but also, as I mentioned, with the ITANC platform. So ELC301 is targeting B-cell lymphoma. And B-cell lymphoma actually already has 3 approved CAR T cells. So you can ask yourself, why even target this indication if there's already 3 approved CAR T cells in this indication and they work so well? Well, we know that they work very well, but we also know from previous studies published in Nature Medicine, as you can see in the figure here, that, so to speak, only 40% have a sustained complete response. And of course, 40% with a sustained complete response is amazing clinical data. But this also means that the majority of patients, even if these CAR T cells are being approved in this indication, are still in need of other treatment alternatives after CAR T cell treatment. And again, just to remind you, when the patients relapse on conventional CAR T cell treatment, they usually lack the target for the CAR T cell. So in this indication, the CAR T cells that are approved are against CD19, and we target CD20. So that's rationale number one. We use another target, not CD19. So we can use our CAR T cell either before or after treatment with the conventional CAR T cells. But then, of course, we've optimized it with the ITANK platform. So we not only target CD19, we believe that we can trigger a second immune response by the patient's own killer T cells against a whole repertoire of different antigens and targets on the tumor cell. So this is the rationale why we go for CD20 and B-cell lymphoma. The hurdles and the risks, the clinical risks, so to speak, are less also in liquid tumors than they are in solid tumors. So this is a little bit more of a niched indication, you can say, for us. And then we have a broader indication with a larger potential in ELC401. We currently are producing CAR T cells according to GMP for 301, and our plan is to initiate a clinical trial by second half of 2022. As I mentioned at the beginning of the presentation, our ambition isn't to take a product all the way to the market, but rather to find a commercial partner that can take over late-stage development and market the therapies. So we see multiple opportunities for out-licensing, and of course the ITANK platform, it's you can say like a base for both 301 and 401, but it can also be out-licensed on its own. 301 is niched against liquid indications. We go initially for B-cell lymphoma. There's an opportunity also in mantle cell lymphoma, we believe. But 401, we believe, is a broader applicability in solid tumors where the greater potential is, also greater risk, of course. But we believe that 401 in brain tumor is a very interesting opportunity. We know that CAR T cells in brain tumors have responded, the patients have responded quite well in this indication. We see an opportunity also in colon cancer, pancreas cancer, head and neck cancer, melanoma, and ovarian cancer, being that the IL-13RA2 that we target is overexpressed most likely on these indications as well. On the oncolytic virus side, the ELC100, as I mentioned, is restricted to neuroendocrine tumors, so it's very niched. In this indication, we only know of one other company that develops an oncolytic virus in this specific indication. We also know that immune checkpoint inhibitors are being studied in neuroendocrine tumors. And for those of you that are not familiar with that product category, it's basically the most relevant immuno-oncological treatment today, you can say, immune checkpoint inhibitors. And they are being approved in more and more indications. And what they do is that they work with the patient's, the tumor's defense of the immune system. So they work very well in a subgroup of patients, but they don't work in the patients that don't already have an active evasion of the immune system against cancer. And of course, CAR T cells and oncolytic viruses, they activate the immune system, so they can be combined with immune checkpoint inhibitors. So we see an opportunity to do that with our oncolytic viruses. 201 is broader than ELC100. It's applicable against most solid tumors, and we haven't chosen our lead indication yet. It's yet to be analyzed. So I mentioned my background shortly in the beginning of this presentation. You can say that I'm an educated biotech entrepreneur. I have a Master of Science in Industrial Engineering and Management with a focus on biotech. And I have a master from Chalmers School of Entrepreneurship, also with a focus on biotech. Ingvar Carlsson is our CFO. He has many years of experience as a CFO for both public and private companies, among other CFO at cell therapy company Idogen. And Magnus Sand is the co-founder and chief scientific officer of Elicera. professor in gene therapy at Uppsala University, previously employed at National Cancer Institute, numerous publications and awards and grants, recipient of these. And as I mentioned, clinical experience from both CAR T cells and oncolytic viruses. And both me and Magnus are two of the major shareholders of Elicera. Agneta Edberg, chairman of Elicera, previous chairman of Immunicum, which is, of course, another immuno-oncology company, so she knows this area very well. She has a very broad profile with over 20 years of experience from life science. She's worked at leading positions at both biotech companies and big pharma companies such as Pfizer. Christina Herder is our, also you can say, a very broad business strategy profile with over 20 years of experience from life science industry. Today, Chief Operating Officer of Medivir, which is another oncology company listed in Sweden. Jan Zetterberg is our legal expert, many years of experience from different senior positions at AstraZeneca's legal department. Margaret Jurvid is our regulatory expert with many years of experience from regulatory and immuno-oncology, and she really knows cell therapies as well, which is very valuable for a company such as Elicera. And Karin Hågendorn, who is our CMC expert. So, of course, these products that we're developing are very complex to manufacture. As I mentioned, Magnus and Dee already have this experience, and through Karin, we also have experience from a person that's been part of actually developing Kymriah, which is a CAR T cell developed by Novartis that is today market approved. So, of course, very valuable for Elicera as well with her competence. So we feel that we have a board with very complementary and relevant experiences. So the share issue that we're conducting now is 62 million Swedish kronor before share issue costs, assuming that the share issue is oversubscribed and we use the 10 million over allotment option. Otherwise, it's 52 million. If we secure the 62 before share issue costs, it should last us until mid of 2023. And the two most relevant milestones that we have ahead of us is finishing the step 1 in the ongoing phase 1/2 study with Advins in neuroendocrine tumors, and also to get an approval and initiate the first clinical trial with a CAR T cell, ELC-301 in B-cell lymphoma. And you can also see here that we have a quite extensive clinical program planned into 2023 and ahead. And I sometimes get the question, can Elicera manage such a broad clinical program, being such a small company? And my answer is, for sure, I think we can. And our ambition is to reach proof of concept in these earlier clinical phase 1/2 studies. But we need to, of course, to prioritize entering phase 2, because having such a broad program with phase 2 studies, I think, is a lot to take on. And how we prioritize in the future after these phase 1/2 studies will all depend on the clinical data that we generate and the interest that we generate from potential partners. And the ITANC platform, we see, being that it's fully developed and there's a big interest in the CAR T cell field, we have as an ambition to try to generate an interest for that platform as soon as possible. So here we don't have to wait for clinical trials. We believe that we can generate an interest before that. So that's what I will be focusing on from the fall and going forward. So again, a short summary of the presentation. So this is a newly launched immuno-oncology company, cell and gene therapy field. We have substantial research grants secured throughout the years, which has enabled us to advance our projects quite far, even though we just turned to investors first time last year. We have an IPO process ongoing. Subscription period ends tomorrow. We have a broad product portfolio with CAR T cells against both solid tumors and liquid tumors. We're developing next generation oncolytic virus. We have an ongoing phase 1/2 study in neuroendocrine tumors. We have a unique approach with our CAR T cells being 4th generation and using the ITANC platform. ELC-201, the next generation oncolytic virus, is quite unique in itself, being that it can elicit 3 mode of actions. We are one of very few European CAR T companies, the only one in Sweden. And the ITANC platform is a unique technology in itself. As I mentioned, we've only identified one other company in Japan that has a similar platform. We have a team with relevant experience, both in management and in board, clinical experience from CAR T cells and oncolytic viruses, a CEO that has experience from immuno-oncology over 10 years, and a board with complementary and relevant experiences. So I'm not sure if I presented this a little bit too long here, but thank you for listening, and I would happily answer any questions you might have.
Thanks a lot, Jamal. Yeah, and that's actually some questions coming in here, and no, it's all right. I think you did it very clearly, and there's a couple of questions regarding the share subscription. You mentioned in the introduction that you're only able to subscribe if you're based in Finland or Sweden. Is that right?
Yeah, correct. So the first day of trading is planned now on Friday, so June 11th. So
yes,
if there's an interest to become a shareholder of Elicera and you are not registered in Finland or in Sweden, then of course you can buy the shares the usual way after June 11th. Then.
And the subscription price is set at 8 kronor. Is that right? 8 Swedish.
Yeah, correct. 8 Swedish kronor. I didn't mention that we're so the offering is actually units. So 1 unit is 1 share and 1 warrant.
Yeah.
And this means that we have the possibility to offer new shares for 11.60 during the period of November 2022. So 2 warrants can give you the right to purchase 1 new share for 11.60 in 2022. And 8 kronor per unit gives us a pre-money valuation of 96 million Swedish kronor. And we're asking for 52 plus 10.
Yeah, good. Well, then there's a couple of questions in respect of the treatments for your products, both CAR T and Oncolytic Virus as well. What kind of treatments do they substitute, Jamal? Could you elaborate a little on that?
Yeah, so it depends on which indication we're talking about. But for the neuroendocrine tumors, if we start there, neuroendocrine tumors is a very heterogeneous type of indication. So it's quite difficult to explain what type of treatment the patients get, because the tumors can they come from the neuroendocrine system, so you can get them in many parts of the body. So depending on where they are and how far advanced they are, you get different types of treatments. But usually it's standard treatments for this specific indication. And it's also standard treatment you can also mention is treatments that inhibit the production of different types of hormones that promote the growth of these tumors. If we look at the B-cell lymphoma field, as I mentioned, we treat patients that are not in first line and not even in second line, but in third line. And these patients, they already can get the conventional CAR T cells that target CD19. And as I mentioned, even though these work very well, the CD19 conventional CAR T cells, the majority of these patients are still in need of other therapies. So that's why we want to go in and treat them with CD19 targeting CAR T cells that have also been optimized with the ITANC platform. So we can target other targets than only CD19 and CD20. And then for 201, we haven't chosen the indication quite yet. And for 401, which is in solid tumors, where we initially go for brain tumor, here these patients have basically no treatment alternatives. It's very, very difficult to treat these patients today. So we know from previous examples of patients being treated with different immuno-oncological products that have brain tumors, that this is actually what we would call an immunogenic type of indication. So for some reason, they respond quite well to immunotherapy. So here we believe that we have a very exciting and interesting chance to treat this difficult to treat indication with our CAR T cells in 401.
If we look into the CAR T cells and your platform ITANC, are you able to actually offer this platform for 2nd generation and 3rd generation discoveries, or is it only 4th generation, because it opens up a much wider market, I could imagine?
Yeah, it's a good question, and the answer is yes. We can offer them to any generation CAR T cell, so we're not restricted to we're not restricted to fourth generation CAR T cells. It's different companies have different ways of using CAR T cells. Some of them focus on manufacturing, some of them focus on the CAR molecule, some of them focus on optimizing the actual T cell. But regardless, the ITANK platform can be used to genetically modify it and include this NAP transgene to elicit this second mode of action through the patient's killer T cells. And just to mention also that it can actually be used for T cell receptors as well, which is another product category. And we use it also for the oncolytic virus. But the patent application for the ITANK is restricted to CAR T cells. We use it in ELC. We use it in ELC 201 also, however, but there we have a unique and specific patent application for that concept to use the ITANK platform also in oncolytic viruses.
So, and then the last question here for your most advanced product, the 100 within OOV. If you succeed, and you will do for sure, thumbs up for that. I know it's complex. We know that it's not always easy, but you look into strategic partnerships. So when you finish the phase 1-2, what kind of companies should we look for in terms of alliances? And I know it's pure speculation, but maybe you can drop some big names. And also, if you have any percentages, what do they normally pay? And of course, it depends on the way you make the cooperation. I understand, but just to get some kind of figures on the table.
Yeah. So are you talking specifically for ELC 100 or in general for CAR T cells and oncolytic viruses?
Well, for ELC 100 and maybe also for the market in general, if you could do that.
Yeah, so for ELC 100, I don't want to mention any company specifically, but I think it's quite easy to figure out yourselves what type of companies these are. I can mention that it will hopefully it will be a big pharma company, and I see it as I mentioned, I see a synergy with the combination with immune checkpoint inhibitors. And more or less any big pharma company that is active within the field of oncology has immune checkpoint inhibitors by today. They've either developed them on their own or they have acquired these types of immune checkpoint inhibitors. So they have these products under development in many indications, and oncolytic viruses and also CAR T cells can be combined with these therapies. So I see a natural and a good partner to be a big pharma company that has immune checkpoint inhibitors under development. Regarding figures and so on, there are a couple of benchmarks. I also don't want to raise expectations too much because these benchmarks are, of course, I mean, they are based on very successful deals and successful product developments, and not all of them succeed, but it still gives you an idea of the value of it. So the most recognized CAR T cell deal, I would say, is acquisitions. I mean, in the amount of almost $12 billion. But this has been the acquisition of CAR T cells that have been very, very close to market approval. So you can't compare them really to early stage CAR T or oncolytic virus development. But you have licensing deals in similar development stages as Elicera is in now, that have generated $40-50 million upfront and $500 to $600 million in different milestone developments. So that's not completely unrealistic if you have something that is actually showing proof of concept in clinical studies. Then there is a big interest for these therapies.
Well, thanks a lot, Jamal. I don't know. There's no further questions, I see. So yeah, thanks a lot, Jamal, for a really interesting presentation, and good luck with the share subscription. And as Danes, we will follow you on the stock exchange, and I know that you will be back in H.C. Andersen very soon to take a deep dive on some of the products, and of course also a follow up on the first day on the stock exchange. So Jamal, all the luck to you, and thanks a lot to the listeners to participate in the presentation. Have a nice day.
Thank you.
You're welcome. Bye-bye. Bye.